Okay. So, hello guys and welcome. So, after mitochondria, like where we talked about the functions. So, in short summary, in brief, we talked about the mitochondrial function, right? We talked about its structure. We now know that at least in eukaryotes around 2,000 copies of mitochondria are present. So, we know that it has got its own genome, right? So, and how the pre-sequences is helping in the protein transfer or the protein transport. Okay. So, keeping those things in mind, now we are going to talk about the chloroplast, and you can see that there are so many similarities with the chloroplast, okay, between the mitochondria and chloroplast. Now, why the similarity? Why are we comparing mitochondria and chloroplast, though they are different organelles, right? Does it mean that in the plants mitochondria is not present? Does it mean like that?
Yes.
Yes.
Endosymbiotic theory, ma'am, because both are double membrane and
Yes.
have a genetic material in them.
Correct. No, so the my question was whether the plant does not have mitochondria. So we know that it is, see, we are comparing mitochondria and chloroplast just because they both are double membrane system. They both have evolved with that endosymbiotic theory. So does it mean that the plant does not have mitochondria totally?
Yes, they don't have.
Are you sure?
Yes.
What about others?
Plants have mitochondria.
Huh?
Plants have mitochondria.
Okay. Rishita is saying no, you are saying yes. How are you going to justify your answers? Rishita, why did you said that the plants do not have mitochondria?
I've never heard of that. And also, when we talk about the genetic material, like mitochondria ka genetic material, I mean, mitochondria has genetic material in case of animals, whereas the same is for chloroplast in case of plants. So, and I've never heard of mitochondria as such in plants.
Okay. Akshaya, would you like to justify your answer?
Mitochondria is present both in plants and animals. Like extra thing is that chloroplast is present in plants, but they're not present in animals. For energy requirement and transfer, mitochondria is needed.
Yes. So it's correct, Akshaya. So mitochondria is present in all eukaryotes. So plant comes under eukaryotic organism, right? So it is also present in the plants. We do compare saying like that because of the mitochondrial chloroplast, but the plant cell also have mitochondria. See, chloroplast is meant for the photosynthesis. Okay, so we know that the animal cells cannot synthesize sugar. It is because of the chloroplast only. So once it is being synthesized and to generate more amount of energy, okay, for all your metabolic activities, for that the energy is coming from the mitochondria. Even for plants to carry out their metabolic activities, mitochondria is required. Okay.
Okay.
Okay. Let's start with chloroplast then. So chloroplast and the other plastids. So we know that chloroplasts are responsible for photosynthesis, okay? And all these things, like this is just the function of the chloroplast, which I'm just saying in brief. All this we will be doing in detail in the biochemistry, okay? So don't worry about it. So chloroplast is responsible for the photosynthesis. Now, both chloroplast and mitochondria, they function to generate metabolic energy. It has evolved by the endosymbiosis. They contain their own genetic system and they replicate by division. Now, when I say they replicate by division, what does it mean is, if suppose this is how a bacteria divides, ये आपका bacteria हुआ, It keeps on growing, and finally it divides like this, right? In the same way, here also, if suppose this is mitochondria, now we have seen like how the proteins and the lipids are transported from the ER to the mitochondria, right? In the same way, it keeps on adding to the outer membrane, to the inner membrane. Okay, so all these proteins will be added in the outer and inner membrane, the matrix solutions, the intermembrane space. Eventually, they will also keep on growing and they divided like this. So this is how even mitochondria are divided or replicated by the division. Same applies for the chloroplast also. Okay. Now, in contrast to mitochondria, chloroplasts synthesize their own amino acids, fatty acids, and the lipid components for their own membranes. The reduction of nitrite to ammonia, it is the essential step that is leads to the incorporation of nitrogen, and that's how the photosynthesis, the main step, your synthesis initiates. Now, since it can incorporate nitrogen to ammonia, that's the beginning step of your amino acid synthesis, right? So जब आपका amino acid synthesize हो रहा है, fatty acid synthesize हो रहा है, so definitely the chloroplast is meant for the food resources availability. This is just the basic structure of the chloroplast. Now here also, in the plant cell, you can see that along with the chloroplast, the mitochondria are present, right? If you see the structure of your plant cell. So here you can see the chloroplast is there. These are your mitochondria. ये आपके Golgi apparatus हो गए. Again mitochondria, nucleus, ER, small vesicles are present. Dots are nothing but the ribosomes. This bada thing, gap, what is it? This one, anyone?
Vacuole.
Correct.
Vacuole.
Correct, it is the vacuole. Okay. And if you see under the electron micrograph, you can see that these are the chloroplasts. Now, how do you understand in the electron micrograph that the chloroplast is present or not? When you see the stacks, okay? What stacks I'm talking here? The thylakoid membrane stack. So if we have got the outer membrane, this is your outer, this is your inner, and then we have got these arrangement of the thylakoid stacks known as grana, right? And then you have got the stroma. So ये हो गया आपका स्ट्रोमा, थाइलोकॉइड, थाइलोकॉइड के अंदर का हो गया आपका थाइलोकॉइड ल्यूमेन. Okay. Sorry. Now the structure and the function of the chloroplast. So we already know that it is double membrane. In addition to the inner and outer membrane of the chloroplast, which is like similar to your mitochondria, the chloroplast is having additional third internal membrane system known as thylakoid membrane. Okay, now these thylakoid membrane forms a network of flattened discs known as thylakoids, and those stacks are known as the grana. Now there are three membranes. So अभी तीन membrane है, तो इसका मतलब the protein sorting will be then of three types. Correct? एक होगा आपका outer membrane के लिए protein sorting. So let's talk about these before we go ahead. Then the figure you guys can explain to me. So one is having the protein integration on the outer membrane. एक होगा आपका outer membrane के लिए, correct? एक होगा inner membrane के लिए. Then you have got for the thylakoid membrane. Now where else the protein sorting is required? Where?
Stroma.
Correct, in the stroma. Then so ये हो गया आपका membrane bound proteins. Let's talk about the soluble proteins, right? तो एक हो गया stroma में, then
Cytoplasm.
अरे साइटोप्लाज्म बाहर है ना क्लोरोप्लास्ट के. This is cell, ये chloroplast. So cytoplasmic protein is different. I'm talking about chloroplast related proteins. So you can have in the outer membrane, inner membrane, thylakoid membrane. You can have protein, soluble protein in the stroma, and where else? So we can have between in the outer and inner membrane के बीच में क्या आता है? The intermembrane space.
Intermembrane space.
Correct. Right? Then what else? Then we have got the thylakoid lumen. So in the case of chloroplasts, the protein sorting of 6 types are needed. Is this clear to everyone? Outer membrane, inner membrane, thylakoid stroma, intermembrane space, and thylakoid lumen. Yes, Krishita, clear?
Yes.
Okay, so I have got one video. Let's check out this video. Are you able to see?
Yes, ma'am.
There is no audio, okay? So what do we observe over here? These are your chloroplasts. So you can see that in one single cell, like so many chloroplasts are present.
Ma'am,
Yes.
where are the other organelles?
I'll come to that. I'll tell you. Okay, that's all. So what we have shown over here is like the, the dynamics of the chloroplast. Okay, let's go back. Okay, so anyone else who would like to give the answer why the other organelles were not seen and only chloroplast was seen? It's okay even if it is wrong, but I just want you guys to, have that thinking ability.
Ma'am,
Because they are pigmented.
Yeah, is it because of?
what? Were they tagged?
Sorry, hello?
Were they tagged
No.
and seen under a fluoropore or something?
No. Tagging means what? You should be able to see that, but we were not able to see. What do we saw? Only the cell
No,
wall,
I mean
right?
the chloroplast.
Yes,
Ma'am,
the chloroplasts only have the colorless, others are colorless.
Exactly. And that's why when they were looking in the under the microscope, everything looked transparent and only the chloroplast was seen as green in color. It's because of the chlorophyll pigment. Rest of your organelles does not have any color. Okay, it can be visualized only with the help of your DIC microscope. What is DIC microscope? No? अच्छा ये वाला सुना है क्या? डिफरेंशियल इंटरफेरेंस माइक्रोस्कोप सुना है कभी? ओके, फेज कॉन्ट्रास्ट सुना है? फेज कॉन्ट्रास्ट माइक्रोस्कोपी?
yes.
Yes, ma'am.
Okay. So what happens is that when you change the phases, now when I say phase change, what does it mean, phase change? So we know that a light is falling on a particular object on a particular see, light travels like this, right? हम ऐसे बोलते हैं light आपका ऐसे travel कर रहा है. You suppose this is one single phase. If I change the phase of another light which is falling on the object, it will be somewhat, you know, कुछ ऐसा भी हो सकता है. If I have another light like this, तो ये क्या होगा? ये out of phase होगा. अगर ये phase आपका 180 है, तो out of phase वाला आपका -180 हो जाएगा. So I'm not gonna see anything. It will be then blank. Okay. Now what had happened here is that अगर मैं phase change करूं, okay, अगर मैं phase change कर रही हूँ, आपका light ऐसे शुरू कर रही हूँ. So what is happening? This phase is 90. कितना out of phase है? वो सिर्फ 90 है. So because of this 90 difference, what will happen? कोई भी object जो रहेगा उसके edges में वो light गिरेगी and वो reflect हो जाएगा. So I'll see something over here, but not over here. Because of this phase difference, then I can say that the other organelles were present. अभी क्या था? In this particular video, what you saw, एक ऐसे खाली cell wall दिख रहा था आपको and directly light गिर रही है. ठीक है? And because of chloroplast is already having its own color, you could see in the white light that it was green in color. Okay, rest of nothing was seen. But if you had noticed over there, ऐसे बड़ा बड़ा gap कुछ था. If you would have noticed those things, किया था क्या notice? ऐसे बड़े बड़े gap थे to which the chloroplasts were not able to move, और एक ऐसे ही बड़ा gap present था वहाँ पे. समझा? We'll come to, if you have not talked about, if you have not studied about the microscopy, then give me some time. I'll start making, PPTs for the microscopy, because microscopy techniques are very important with respect to the exam, and we have got many. Confocal is there. A phase contrast is there, so you must have talked about the bright field and the dark field. Bright field वही होता है जो आपका normally हम compound microscope में देखते हैं. Bright field हो गया, dark field हो गया, phase contrast हो गया, confocal हो गया. Then we have got TEM, transmission electron microscope. Then we have scanning electron microscope. Right? अब तो बहुत आने लग गए हैं, but we'll go with all these basics वाला. ठीक है? Now, let's talk. I'll just give you a little more reference of like how the phase contrast microscopy is happening. See, इतना अगर आपको मैं बोल रही हूँ that you have got the object over here. ठीक है? यहाँ पे आपका specimen present है. Let's say this is your particular plant cell. ठीक है? So obviously on the object, this is your object. On the top of the object, you have got the objective lens. राइट, ऑब्जेक्टिव लेंस रहता है. ऑब्जेक्टिव लेंस के ऊपर आपका लाइट आता है, जहां पे लाइट सोर्स रहता है. करेक्ट? नाउ इन द केस ऑफ द फेज कॉन्ट्रास्ट माइक्रोस्कोपी, यू हैव गॉट समथिंग कॉल्ड फेज प्लेट. ओके, नाउ व्हेन लाइट इज फॉर्मिंग, सो यहाँ पे क्या होगा, हम लोग 2-3 लाइट डालेंगे. तो एक लाइट क्या करेगी आपकी, ऐसे जाएगी. ओके, जो यहाँ पे जो आपका क्या कहते हैं उसको, अपर्चर. ओके, सो वी आर गोना एडजस्ट दिस अपर्चर जहां से आपके लाइट पास होगी. सो ये आपका स्पेसिमेन में गिरेगा ऐसे. ओके, देन आई एम हैविंग अनदर लाइट विच इज आउट ऑफ फेज. तो जो आउट ऑफ फेज लाइट रहेगी, लेट्स ड्रॉ इट समथिंग ऐसे. ओके, ये कुछ यहाँ पे स्पेसिमेन में गिरेगी. ठीक है? नाउ ऑल दीज व्हेन इट इज गोइंग टू गेट कंडेंस, आपका मालूम है यहाँ पे कंडेंसर लेंस रहता है. नाउ डिपेंडिंग ऑन दैट इट इज गोना फॉल and I'm gonna get all those answers in the image I'm gonna get. Okay, did you get it?
Yes, ma'am.
Yeah. मुझे कुछ गड़बड़ लग रही है, 1 second. ओहो, सॉरी, एक छोटा मिस्टेक हुआ है. I have to explain it again. थोड़ा सा गलती हुआ है in the alignment. Okay, I'll start with the first light source. Okay, light source हुआ यहाँ पे आपका. उसके बाद you have got your, यहाँ पे सॉरी, कंडेंसर लेंस पहले आते हैं, because they need to bring all those light, आपका जो light source होता है उसको एक जगह पे फोकस करना है, right? That's why you need the condenser lens. Then comes your specimen. तो यहाँ पे आपका specimen आ जाएगा. Okay, now this light which is falling on it, so you have got those phase plate which is helping in bringing the light in the different phases. फिर आपका ऑब्जेक्ट है, ऑब्जेक्ट के बाद ऑब्जेक्टिव लेंस रहता है. ओके, एंड देन यू हैव गॉट दोज इमेज फॉर्मिंग lenses are there. Okay, so the difference is that in the case of the rest of the microscope, in the phase contrast microscope, you just have got the phase plate. Okay, so this phase plate will bring your light in different phases, because of which you will be able to see. Okay, anyways, अभी के लिए छोड़ देते हैं. I don't have the proper image and I'm really bad at drawing. So let's not get confused, and I'll start with the microscope also, the techniques. Okay, before that, I remember that we were doing electrophoresis. I remember now. I completely left that. Anyways, we'll come to that topic separately. So the outer membrane of the chloroplast, it contains porins similar to your mitochondria, and it is therefore freely permeable to small molecules. Again, so it means the intermembrane space is going to have the similar composition with the cytoplasm, which is like similar to your mitochondrial case, right? In contrast, the inner membrane is impermeable to ions and metabolites, and therefore able to enter chloroplast only via specific membrane transporter. So this scenario is totally similar to your mitochondria case. Okay. Now, in the mitochondria, so your ATP synthesis is happening like this, where the complexes are present in the inner membrane and ATP synthesized in the matrix. Now, similar thing, instead of the inner membrane, it is the thylakoid membrane where it is present and the ATP is synthesized. Okay. Now, where the ATP synthesized ATP is going? It is going in the stroma. यहाँ पे सिर्फ एक ही स्टैक दिखाया उन्होंने thylakoid का, right? So after the synthesis of ATP, it is going in the stroma. Is this much clear? This is simple, right? Now let's talk about the chloroplast genome. So like mitochondria, even the chloroplasts contain their own genetic system, which shows their evolutionary origins from the photosynthetic bacteria. So if we see that they have got their own RNAs and tRNAs, ribosomal proteins are there, they do have their own RNA polymerase subunits. Then of course the photosystem 1 and 2, cytochrome BF complex, all this which is specific to your chloroplast, right? Because chloroplast is involved in your photophosphorylation, resulting in the photosynthesis. Okay, it also have proteins which is involved for the metabolism, protein folding and membrane insertion. This membrane insertion means what? They are the protein transporters which is helping in the integration of the protein from your which is meant for your both nuclear as well as chloroplast genome proteins, जो आपके outer in thylakoid membrane इन सब में आपका protein insert करता है, ठीक है? Now let's talk about the protein sorting of the chloroplast. So 95% of the chloroplast proteins are encoded by the nuclear genes. ठीक है? बाकी बचा कुचा is by the chloroplast. Now, as in the case of the mitochondria, we had pre-sequences, correct? Now here we are calling those signal peptide as transit peptides, which is around 30 to 100 amino acids. Okay, pre-sequences क्या था आपका? 15 to 55 amino acids. Okay, Now, in the case of chloroplast, it is your transit peptides of your 30 to 100 amino acids. Now, these are the responsible signal which will direct the protein translocation across the two membranes of the chloroplast. So, this is something which you have to remember. Okay. Yes. Now, who would like to try giving the explanation for this? Akshay, would you like to try explaining? It is really very similar to the mitochondrial translocation, protein translocation. The difference comes in over here, like instead of Tom and Tim, we have got Talk and Tick. Yes, would you like to try?
Yes, ma'am. So there is a peptide, there is HSP70, like it makes sure the protein is not folded, and it goes to the TOC complex. Before going that, there is ATP hydrolysis and GTP hydrolysis. There is also a transit peptide along with it, and it passes through the TOC complex. And when it reaches the TOC complex and TIC complex joins, and it allows the peptide to pass through the intermembrane space, and it comes out of the TIC complex, and there comes HSP 93 and SPP. So there is again hydrolysis, and it pushes the peptide to the stroma, and if it is to the membrane, it pushes towards the membrane.
Okay,
Yes.
good. Yes. So, in addition to what she has explained, so transit peptide is already present, okay, in your newly synthesized polypeptide chain. So this transit peptide is giving a signal, okay, like this should be transported to the chloroplast, okay. So this is the signal that these proteins should be transported to the chloroplast. Now, once it enters, in order to open the translocator, you need ATP, GTP hydrolysis. Now, in the case of chloroplast, there is this additional hydrolysis is required, whereas in the case of mitochondria, it was only once. Okay. Now, so it is passing through the TOC complex, then it is passing to the TIC complex. Now, what is additional over here is that ATP hydrolysis again for the opening of the TIC complex. HSP93 is required in order to pull the polypeptide chain out from the TIC complex. Okay, and SPP is again, if you remember, we have always talked about the signal peptidase, right? So we need to remove the signal sequences. For that, it is the signal peptidase. The signal peptidase हम बोलते हैं ER में, right? In the case of the mitochondria, it was MPP. Now, in the case of chloroplast, it is SPP, which stands for your stromal processing peptidases. Correct? वहाँ पे MPP था, matrix processing peptidase. Here it is stromal processing peptidase. Okay, which is present in the case of your chloroplast. Now, once this is being done, HSP70 again takes this protein and keeps it in the unfolded form unless until it is required to be completely folded. A complete folding कहाँ पे होगा आपका HSP60 में ही होगा. Okay, is it clear to everyone else? The role of HSP93, SPP70.
Yes,
Okay.
that's fine.
Okay, so now next. So import the proteins into the thylakoid lumen and the membrane. Okay, so here we see that we have got the three different ways by which the proteins can be imported to the thylakoid lumen as well as the membrane. Out of all these three pathways, the TAT pathway is important with respect to the exam. Okay, so many a times they have asked already about the TAT pathway. So this TAT pathway, TAT stands for twin arginine translocation. Okay, it is your twin arginine translocation pathway. So what does it mean? It means that it has got that sequence Twin arginine means 2 arginine residues will be present, okay, at the end terminal of this protein, which is supposed to be transported or translocated with the help of the TAT translocon. Okay, now what is happening in the TAT pathway is that this twin arginine will be recognized, okay, that will be brought to your TAT translocon. This is your TAT translocon, and now this will be taken inside. Now what do we see here additional? What do we see here additional? That it is also resulting in the proton gradient. So along with the protein, the H plus are also going inside, right? Did you see this H plus are also going inside? So because of which what is happening? The lumen, the pH is dropping, right? Later, this twin arginine signal is being cleaved by the TPP. अब TPP क्या है? वहां पे क्या था? Matrix, stromal. So T stands for your thylakoid. So thylakoid processing proteases or the peptidases, which is actually removing this signal sequences. Okay, is this pathway is clear? TAT pathway. Okay, now next one is your SEC pathway. So SEC pathway same है जो हमारा ER में हमने पढ़ा था. So in the case of the SEC pathway, HSP70 है, it brings over here to the translocator, and ATP synthesis, ATP hydrolysis happens in order to open the translocator, and then the proteins are getting inserted either into the membrane or into the lumen. Okay, then TPP again removing your signal sequences. Simple. Now coming to the SRP pathway. So in the SRP pathway, what do we see? Again, see, this is like the translation was happening. Okay, now once the translation, this is your co-translation translocation, if you remember. Along with the translation, the translocation is also happening. So SRP is there. So in this case, it is your chloroplast signal recognition particle, which will recognize this sequence, which is present in the newly synthesized polypeptide chain. The HSP70 is there. Now, this is all brought to the translocon known as ALB3. Okay, and then it will be inserted into the membrane. Clear? All the three pathways are clear. This is only for the import of the proteins into the thylakoid lumen as well as the membrane. तो उसके लिए हमारे पास 3 pathway हो गए, TAT pathway, SEC pathway and SRP pathway. Okay. Yes, Tanvi, is it clear?
Yeah, ma'am.
Yeah. It's simple. So you can just,
Ma'am?
yeah.
So your co-translation as well as post-translational tagging both happens?
Yes, yes. In the case of your thylakoid proteins. Why? कैसे possible हो रहा है? Because the chloroplast has got its own genome, right? So they have their own protein. So chloroplast is meant to synthesize your photosystem 1, photosystem 2, responsible for the photosynthesis to take place, right? So that is possible with the chloroplast genome. तो वो क्या करेगा? वो अपना ही ribosome, अपना ही RNA, tRNA लाएगा, translation करेगा, and then this will be inserted into the thylakoid lumen. So if you remember that photosystem 1 and 2 कहाँ present है हमारे, right? Thylakoid membrane में present है. So all these will be brought with the help of the SRP pathway then. Yes?
Yes, ma'am.
Okay. See, अगर फिर भी कभी doubt आ जाए in future, like not able to understand with the help of the recording also, so you can just see, you can understand from the figure that this is the 13th chapter, and as I mentioned that it is from the Cooper. Okay, so the figures are directly taken from the Cooper. Again, 13th, it means it is a 13th chapter which is going on. The same thing which we just discussed now. So I'll just go through in case if I miss any point. So the twin arginine translocation of the TAT pathway, it is related to the bacterial membrane translocation system. In this pathway, The proteins are recognized by the twin arginine signal sequence and transported across the membrane in a fully folded state. The second pathway, sec pathway, which is related to the pathway used to translocate the proteins into the ER, as I mentioned. So in this pathway, the thylacate signal sequence is recognized by the sec-A protein. वहाँ पे क्या था आपका in the ER? It was sec61. Okay, the translocator का नाम जो था. यहाँ पे है आपका sec-A. Now, the proteins with the transmembrane sequence can exit the sec translocon laterally and can be incorporated into the membrane. Finally, there are certain proteins which is incorporated into the thylacate membrane via the ALB3 insertion, which is related to OXA1 in the case of the mitochondrial inner membrane. Remember, so जो mitochondrial membrane proteins है वो आपके OXA1 translocator से जा रहे थे. Similar to that, in the case of the thylakoid, which is specific to the chloroplast, it is your ALB3 translocon. Okay, so now these proteins are targeted to ALB3 by the chloroplast signal recognition particle, which recognizes the proteins encoded by the chloroplast genome, okay, and translated by the ribosomes in the stroma. So these are the three pathways for your thylakoid, lumen, and the membrane protein insertion. Yes. Any doubts over here? Okay, so I'll be stopping here now.